This is Audiomunity, a podcast about our body's never-ending fight with the outside world. Hi everyone, this is Audiomunity, recorded on May 20th, 2025. This is episode 40. I'm Kevin Bonham, and I'm joined today by Matt Woodruff.
Hey, everybody.
And I'm not going to do a silly this episode brought to you by. Stuff seems way too bleak for that right now.
I'm going to keep doing it. And
I would really like, I picked this paper in part because I feel like it barely touches on any of the BS that's happening right now. I was thinking yesterday, like, imagining listening to these episodes in like 5 years and thinking, like, how unhinged are we going to seem? Or is it going to be like, oh, yep, that was prophesizing the future and everything is terrible?
You know what they say about prophesizers? Usually right.
Yeah. I've been recently rereading a bunch of like, there's this whole genre of novel that are like Greek myths retold from different perspectives. And I recently read one called 1,000 Ships that's like the retelling of the Trojan War, but from the perspective of all the women involved. And Cassandra is like this side character. So for those of you that don't know, Cassandra is a woman from Troy who was basically cursed by a god to have the gift of prophecy, but no one believes her when she speaks. So she's like, she has perfect predictive power, but no one will actually listen to her. And so the whole thing talks about, like, it's never from her perspective directly. It's like all the people around her. And they just like treat her like she's a mewling, like crazy person, just like talking nonsense constantly. I kind of feel like that sometimes. Like, I've stopped talking to my family about this stuff because it's like they just think I'm overreacting, you know?
Right.
That, like, could it really be that bad? Like, I'm not reading about this constantly in the news. And it's like, no, actually, like, my whole career is over. But anyway, it's not that bad. It's just my whole career.
Hot take. I don't believe you read. I believe you watched the hit Netflix show Chaos and are now repurposing. What do you think?
What is Chaos?
That was a good catch.
No, I don't actually know what Chaos is.
No, I obviously do believe that you read. I know that you ran a book club for quite some time, but no, there's a cool Greek mythology in modern times show that is a pretty fun watch called Chaos. It's a little, yeah. Just go take a look.
Okay, sounds good.
That's my quick Netflix plug. I'm taking Kate's role today.
Yeah,
as
exactly.
The person
We're
that has
trying to
watched
get some
the
pop
TV
culture
show.
in here.
Exactly.
Highbrow pop culture.
Yeah.
Resetting for a moment the paper that we're going to read today. Yeah. Matt, can you guess why I chose this paper?
Well, I think we're on a real tolerance kick here at Audiomunity. I think that we have talked a lot about what immune tolerance is and what how the microbiome plays into that, and how immune surveillance kind of works. So my guess is that it sort of sits at the intersection of a lot of those spaces where, you know, we're sort of talking about, you know, at these barrier environments, how is the immune system balancing itself with its interaction with the outside world, the potential to respond to something that's aggressive, but also the need to constantly say, hey, chill out, bro. It's kind of fine.
Yeah, so that is a very kind guess. It gives me a lot of credit. Actually, what happened is I pulled up Science Immunology. I put in the search bar spatial transcriptomics because we are trying to write a grant on spatial transcriptomics, and frankly, it is very hard to write grants in this environment. And I thought, you know, can I use Audiomunity to do something that I should probably be doing anyway, which is reading about the ways that spatial transcriptomics is using? And this popped up, it had immune privilege in the title, and we've talked about immune privilege recently, and I was like, okay, that sounds good enough. But that said, it does kind of connect with current events in the sense that this is, after reading it, I mean, I didn't pick it for this reason, this is the technical term is fuck ton of mouse work.
Yep.
Like this paper has so many different mouse models, so much work on like really dissecting the details of this system, which is quite cool. Completely impossible. I mean, this would make no sense to do in an organ on the chip or whatever hell other things that NIH is going to promote now. In lieu of mouse models, but it seems like very cool. And at the end, it uses spatial transcriptomics on humans in a way that, like, if you hadn't done the first 6 figures of this paper, the spatial transcriptomics would make no sense, and they would have had no idea what to look for.
Yep.
But cool use of the technology anyway. So the paper itself is called "Regulatory T-cells in Skin Mediate Immune Privilege of the Hair Follicle Stem Cell Niche." So it's just first author is Jarish Cohen, and the corresponding author is Michael Rosenblum from UCSF. And I don't know, like, top line, the title is pretty clear. Looking at Tregs in skin using a pretty cool model system. What did you think of this paper?
I like the idea of mouse Mohawks, first of all. I'm sort of excited about...
I kind of picked the title ahead of time. I feel like Mohawk Mouse is going to be going to be the title here.
No, I think it's a cool application of like slightly deviated methodology, right?
Yeah.
Like, you know that I'm all about location, immune location, and controlling things into environments. I think they do a really good job here of thinking through, generally how to isolate the effect size or the effect that they're seeing to, a very specific area. No, I think, it's what happens when you really think about a model and you take the time to, develop something that's slightly new and then, use that to great effect. And like you said, they use a lot of different mouse models to dissect the different components of what are needed, for example, for a Treg to function in different environments, which I think is really cool. It's also not the kind of science that I, to be honest with you, ever hope to do. Like, it is well outside of where I want my mouse colony to be. But
the thing that I really liked about this is, like, I feel like they used very, very standard models. They used a bunch of different models in very standard ways, except for like one little tweak that really just makes the whole paper. And so let's get into that. First, I do want to say that the intro, I thought the first like paragraph and a half are like a really nice summary of like probably 3 weeks of a graduate level immunology course in terms of tolerance, right? So they talk about things that we've discussed on the podcast. Central tolerance is when T cells and B cells are developing. During that period, there are mechanisms to weed out the T cells and B cells that have rearranged their receptors in ways that are autoreactive. So in T cells, it happens in a special organ called the thymus. We're going to be talking mostly about T cells here. B cells, it happens in the bone marrow, but similar strategies where activation during development gets you the axe. They talk about peripheral tolerance, which they're sort of... the way that they are describing peripheral tolerance here is really happening in secondary lymphoid tissues like lymph nodes and spleen, where as naive cells are patrolling, if they encounter stimulation in the absence of inflammatory signals, that is a signal that they are autoreactive. And so those cells get deleted or shut off effectively through energy. And so that's sort of like the paradigm that they are talking about. And one of the things that they describe as sort of like the main gap that this paper is trying to fill is that most of the ways that people have been able to dissect peripheral tolerance, which is largely mediated through T regulatory cells, is through essentially systemic knockouts, right? You do some kind of genetic manipulation. Again, these models we've talked about before, where you say under the promoter that is the transcription factor that is indicative of T regulatory cells is a promoter called FOXP3, or is a transcription factor called FOXP3. So under the control of a FOXP3 specific promoter, you can do things like express diphtheria toxin receptor, and then you treat with diphtheria toxin, you eliminate Tregs. But that, when you're doing it under the FOXP3 promoter, you're doing that systemically. Mm-hmm. And so if you're wiping out Tregs throughout the body, you're doing it in lymph nodes, you're doing it in spleen, you're doing it in bone marrow, presumably. Like, you're just wiping it out everywhere. And so the effects you see are systemic breaks in tolerance, which makes sense.
Which is useful because there are, as they mentioned in the introduction, there are actually human diseases that reflect that kind of systemic disruption to the Treg population, right? So we don't want to make it seem like, these studies have been sort of generally disconnected from real human immunology. We've learned an enormous amount from those studies.
Yeah, 100%.
But also, location is important, right? Space-specific effects are important. So I completely agree with you. It's really a really cool way to sort of twist these questions towards, how are these things acting in local tissues?
Yeah. So the model that they actually construct is sort of a combination of a couple of different things. So one of which is this tamoxifen inducible promoter system. So the idea here is that there's a chemical called tamoxifen that when you treat with it, it induces essentially a gene rearrangement, right? So the specific gene rearrangement they're doing is they're knocking in a fluorescence reporter, and they're are they also doing diphtheria toxin? They're doing some other way of deleting the thing. So Rosa 26, lock stop, the D tomato, FOXP3, and
I think at the beginning they're just asking what gets.
They're just putting in the They're just putting in the fluorescence marker.
It's the tomato. Yeah.
So they're basically just putting in a fluorescence marker that when you treat with tamoxifen, it's not like the tamoxifen is not causing the expression. What tamoxifen is doing is it's causing a Cre recombinase genetic manipulation, so those cells are permanently now labeled with this fluorescence reporter D tomato. Right. So it's turning on expression of this gene in a way that is going to persist after the tamoxifen is taken away.
Yep. And interestingly, and I didn't know this actually. Interestingly, it's not tamoxifen itself. Yeah. And I honestly had never heard this. So you inject tamoxifen, right? Well known.
Yeah.
What I did not know is that it's not tamoxifen that has the active effect. It is a metabolite of tamoxifen after tamoxifen hits the liver.
Right.
And that is this compound 4-OHT.
4-OHT. Yeah.
4-OHT that they are then going to use topically later on. But that is an interesting quirk of this system. That
I had no idea as well.
Had never seen. So anyway, good on them for reading the literature.
Yeah, exactly. So treat with tamoxifen. Yeah. It gets converted by the liver into, so you inject it intraperitoneally, so you basically into the body cavity. So it goes to the liver, it gets converted into this 4-OHT, and that's systemic. It goes systemic. So basically all of the Tregs, because it's still under the control of this FOXP3 promoter, so all of the Tregs systemically turn on this fluorescence protein, so they can track them. And in Figure 1, they show that very nicely, like this, just like the rest of the literature you'd expect. You get all of your Tregs labeled in all of your draining lymph nodes, in all of your secondary lymphoid tissue, and in the skin. You get fluorescence reporter activity. But then what they do is they take this 4-OHT compound that is actually the active compound, and they think like, well, how can we get this just in a site that we care about? They're focusing on skin here. I don't know. I didn't look at the literature that these people have published previously to know if skin immunology is something that they have previously cared about. But obviously skin is very accessible. So they're going to use skin as a model of local effects. Yeah. And they basically just paint on 4-OHT in acetone, it gets absorbed into the skin, and then they want to ask, do you get a local effect? And it's cool because they do this Mohawk treatment that you were describing, where they basically shave. For those of you that are looking, I'm trying to use my hands to show, but basically they shave on the back of the mouse. They shave both sides of the back, and they leave a strip of hair in the center.
Yep.
They paint the tamoxifen 4-OHT on one side, and they paint the other side with just acetone, with vehicle control, essentially. Mm-hmm. And then they can look in the skin where they have painted, and then they can also, later, they can... they use the term harvest. It was giving me very like Silence of the Lambs vibes. Like after skin harvesting of these mice, they can look really closely in the skin, and they can also look in the draining lymph nodes on either side of the mouse. And then they can, of course, look in other secondary lymphoid tissues as well. And they basically show in Figure 1 that the promoter activity, the turning on of this fluorescence protein, is really only happening in those Tregs, excuse me, that are present in the skin on the side that they have painted the 4-OHT onto. So this is really nice control, very clearly showing you get really high expression of cherry, and I think they said like 70 to 90% of the T regulatory cells in the skin on the side that they're painting it on, and you get none in the side that they don't paint it on.
Yeah, and I think, just sort of review of how that lymphatic structure works, and it's, it's fairly similar in mouse and humans. It's obviously a lot more complex in humans, but there is sort of bilateral symmetry of your lymphatic system, right? So it sort of runs down your spine, and this is true in mice as well. And when you separate across that central line, there really isn't much contact between the two sides of the lymphatic system, right? So in a mouse, you can think about everything sort of draining up towards the head, right? So your lymphatics and lymphatic capillaries, the smallest vessels, sort of start in the feet, and they drain from everywhere. But basically what they're doing is they're sort of ascending up the mouse, and then they're eventually converging into the thoracic duct. But on the way, they're running into all of these lymph nodes, right? So if you're painting on one side versus the other, you really have no mechanism for that chemical that you're painting, to get to the other side of the mouse. There's nothing connecting those two things except for the blood. So you're not going to see really any transfer of any of those chemicals that you're adding, unless you've added so much that it finds its way into the blood, gets into the spleen eventually, because the spleen is filtering the blood, and then starts leaking out into the surrounding tissue. So it really takes a lot of drug in order to get past that sort of bilateral symmetry.
Yeah, unless somehow, the lymph was like, or the chemical was like passing the wall of capillaries or something. Mm-hmm. And that's like part of the point of lymphatics, is that just doesn't really happen. Like the fluid that is in the tissue, the way that fluid gets back into the blood is through the lymphatics, through the sort of typical anatomy. It's very hard for cells to get from tissue into the blood through the walls of capillaries.
Correct. They're designed for that. I would say, the counter argument to that, or the thing to keep in mind in that, is that the reason that lymphatics exist is because those capillaries at some level are leaky. You lose about 10% of your blood volume per day to the surrounding peripheral tissue. And that's important, right? That's how your cells get fed, basically, by the blood vessels.
Right.
So you really are thinking about the lymphatic system as sort of a recycling system for all of that lost fluid. But yeah, from a cellular perspective, very, very hard to go get into a capillary. And also the fluid flow is such, right? The hydrostatic pressure.
That's what I was going to say.
Yeah, is such that it is one directional, right? That fluid isn't just naturally passing back and forth into the capillaries. It's coming from the capillaries and pushing out.
Turns out your blood vessels are under some significant pressure.
I wonder what does that?
I don't know. I'm sure there's something. I'm sure some people have looked at that. It's the heart, by the way. Yes, thank you. Audio effects here on Audio Immunity.
You're welcome.
All right, so Figure 1 is basically Figure 1 is just showing like the system works. It works as expected. And like I said, it's this clever sort of combination of it's a sort of standard model with this one little tweak, this idea that you're painting on the 4-OHT rather than treating systemically with tamoxifen, that you can induce local expression in just the skin tissue under a FOXP3 promoter, whatever you want. In figure 1, it's a fluorescent reporter. They do a little bit of like lineage tracing. They show that the Tregs in the skin don't really go to other places. They show that they persist in the skin for actually like pretty decently long period of time. Yes. They're still detecting some at 94 days, which in the life of a mouse is pretty long. They say somewhere something along the lines of like these cells are like intrinsically able to persist or something, like it's something special about those cells. Like it could just be stochastic, like, you know, some cells die at some rate and there's a half-life, and they didn't say anything like this is not necessarily something special about those cells, but that was my minor quibble with figure 1 was the clearly descending line towards 0 and then making the statement that some of these cells seem to be able to persist in the skin. It does feel like there's probably just a half-life there and a stochastic drawdown. That said, 94 days is a long time. So let's leave that aside. So these are fairly long-lived cells. So then they, in figure 2, they want to see what is happening under inflammatory conditions. And so I think this is where they start with another thing that I like. There were a couple of things as I was reading through the methods here that just like struck me as, so they're using tape striping or tape stripping. So basically they're putting tape on the skin and peeling it up to cause what they call like low level, like slight damage, to just cause a little bit of just like basically piss off the mast cells in the skin and induce some inflammation, right? You get some histamine, you get some TNF, etc. Because you're just putting tape on, you're ripping Band-Aids off, essentially, on these mouse. That's exactly what you're doing. You're putting a Band-Aid off and ripping it off. And here they're again comparing the painted on 4-OHT with systemic tamoxifen induction, and they're trying to look like what's happening to those Tregs that are expressing the fluorescence reporter. So they can track them. When they harvest the skin, precious, they are then able to look. Sorry, I thought I was going to let that slip through. So
it caught me.
They're able to look at the where the Tregs are, right? They can do flow cytometry and sort on all of your typical markers plus the tomato fluorescence protein. And what they see is kind of what you would expect. You get some induction of inflammation. It's pretty similar in both the 4-OHT and the tamoxifen-treated cells. When you only paint, you don't get, the Tregs aren't like going to other places. They're hanging out specifically in the skin. They're not going into the skin draining lymph nodes. You're getting induction of inflammatory cytokines the way you would expect. Like all of this stuff is happening sort of as you would expect. And one of the things they wanted to do is they wanted to see, like, is the inflammation or is the suppression of these T regulatory cells, are they suppressing CD8 T-cells specifically? And so they do an anti-CD8 treatment where they basically wipe out the CD8 T-cells, and basically, like, it does reduce the inflammation, but it's not completely gone. So what they're saying is that the effect of these T regulatory cells is to suppress inflammation generally. Some of it is CD8 mediated and some is not. And now I'm just trying to remember, are they doing the diphtheria toxin here yet? Because
no, you've got it. I mean, you've got diphtheria toxin administration right up at the top of figure 2A. Yeah, so what you're definitely ablating.
Oh, you're right.
Local versus systemic ablation of these cells. Yep. And so I think what they're looking at is, so you're putting out the local DTA versus systemic DTA. So B, you're looking at the size of these lymph nodes and basically seeing that if you ablate Tregs systemically. Unsurprisingly, perhaps, you get enlargement of all of those draining lymph nodes. Interestingly, if you do the 4-HT, even though they showed you that they can ablate or they can target those cells in the previous figure, you're not getting the same kind of generalized inflammation within the draining lymph nodes, right? So I think you're seeing the ablation effects locally and not necessarily systemically in the same way. And then we've got, some effect, it looks like, on, skin CD8 T-cells, for example, in H after you do some taping. And, interestingly, the folliculitis, I think, is really what caught my attention. So, what you're looking at in I is basically that, even just local ablation of these Tregs and not systemic ablation is capable of inducing sort of this uncontrolled inflammatory process they call folliculitis here. And that really was sort of the take-home for me. It does not take a systemic ablation of Tregs in order to get the sort of inflammation that leads to obvious pathology in the skin. So
yeah, exactly.
These cells are doing something.
Yeah, they're clearly suppressing stuff. When you rip off a Band-Aid, and it hurts and the skin is red. The reason that you don't then lose all your hair and all of your pigment cells is because you have these Tregs hanging out around the follicle that are suppressing ongoing inflammation.
Thanks, Tregs.
Thanks, Tregs. We love you. You're doing great. All right, so in figure 3 now, what they do is they do what I feel like is now sort of de rigueur in this kind of immunology paper. Single cell transcriptomics. Take out all the cells. You the technology is very cool. We're not going to go into it, but basically they're able to do RNA sequencing of individual cells, so you can see what the expression pattern is on individual cells. They're able to sort the cherry expressing Tregs compared to other cells, and they're able to compare them. And they're looking in this, what they're comparing now is they're comparing the Tregs that come from the skin draining lymph node versus Tregs that are coming from the skin, and looking at differential expression patterns. They make a big deal of this. I feel like this figure is like the hypothesis generating figure for the rest of the paper. They basically are trying to identify, like, is there something special about the skin resident T regulatory cells that are affected when you are doing the skin painting compared to when you're doing systemic administration and you're just looking at the skin draining lymph node for T regulatory cells. And they say that basically you get a strong regulatory phenotype. They make a big deal out of a couple of genes. And the various clustering, the various clusters that they see comparing these two populations. The thing that jumped out at me is the homogeneity of the UMAP on this figure. Like there's two kind of clear blobs, and then they identify other clusters that, at least on the first two axes, are not particularly well distinguished, but nevertheless they sort of identify a bunch of additional clusters, and they show sort of blocks of regulatory genes, and they say that this regulatory phenotype is especially expressed in the T regulatory cells that are in the skin compared to the T regulatory cells that are in the draining lymph node. The first thing that they pick out, sort of interestingly, is CTLA4.
Yeah.
And so CTLA4, like if I was doing this experiment and I saw CTLA4 jump out, I'd get really excited because CTLA4 is a gene that a lot of people have looked at previously. It's a very well-known suppressive gene that is used to essentially downregulate other T cells. I reminded myself of what CTLA4 does at some point.
It inhibits signaling through the T cell co-receptor.
Right. That's right. So it blocks the co-stimulatory receptors so T cell signaling, back to first year immunology. you have the T-cell receptor that binds to MHC, but you also need co-stimulatory molecules. You have the co-receptor CD8 or CD4, but then you also have these co-stimulatory molecules that are upregulated on antigen-presenting cells in the presence of inflammatory signals, right? So when the innate immune system gets its signals, it basically turns on co-stimulation.
And now
the co-stimulation is needed to not become anergic or get deleted during development.
What is that protein?
CD8086?
Yep, and the receptor is? What's on the T cell?
I don't remember.
CD28.
CD28. Okay, thank you.
Yeah, no, so, so the...
That sounds vaguely familiar.
Yeah, exactly. Yeah. I recently had to do this because we're talking about like making a B cell do some of these things, and so you've got the T cell receptor and you've got CD28, right? And so the T cell receptor engages the MHC. Usually CD28 will engage CD80 and 86, but CTLA4 acts as a different binding partner than CD80 and 86, except when CTLA4 binds, CD28 does not signal. It actually prevents the signaling of signal 2.
So it's a higher affinity receptor that essentially blocks CD80 and 86 from being able to bind.
Yep.
So thus you don't get second signal, thus you don't continue to signal, and you get suppressed. So I was just imagining, so figure 4 is like, I'm just imagining being the graduate student or postdoc that is like doing this experiment. They went through the trouble of making this mouse. So they basically like made a mouse where they could delete CTLA4 because they're thinking like these special Tregs, they're doing suppression. They're really upregulating CTLA4. Like this is our mechanism of suppression. Man, this is going to be awesome. So they make the DTR mouse. They delete CTLA4. Everything's going great, and it does nothing. Figure 4 is just like nothing. CTLA4 does fuck all. Sorry, guys.
Yep. No, it's rare, I would say, that a paper at this level has an entire figure dedicated to the fact that something that you did didn't work.
Yep.
And it really speaks to the amount of effort that must have gone into this.
Oh, God.
Because there's an editing process at the end of all of these papers, and we've talked about it before, where like you go through all of it, there's this huge narrative that gets built, right? And at the end, you sort of, what's the quote? You have to kill all of your babies, right? You have to like disengage from the work that you did and tell the best story you can with the data you have in front of you. And usually when something just falls right on its face, you just kind of like
supplement at best, right?
You
like
push it out and you say, "Yep, look, CTLA4 was highly upregulated. We made the mouse and it didn't work." So
yeah, it's one line. It's despite the fact that CTLA4 was highly upregulated,
right?
It was actually not necessary. See figure supplement 35G, and then it's done. It's
pain.
Matt is showing the figure. Yeah, it's a giant figure. Panels through eye, showing all of the ways in which CTLA4, despite looking really great. I mean, you have to imagine this was at least a year of work to generate this figure.
And probably in parallel, I imagine what they did was they took a couple stabs at different mechanistic possibilities, and
I don't know. If you saw this signal from CTLA4, that would be your first like. I would have been banking on CTLA4 personally. Like you see that signal. I mean, it's the most highly upregulated gene. Comparing, I mean, CTLA4 is already highly upregulated in lymph node.
And it's known Tregs, that it has an effect there, right?
Right, and so if you see that your skin Tregs are expressing even more CTLA4, and not only that, it's the most differentially expressed gene. Like, I'm making that mouse, and I'm thinking I'm taking that to the bank. And then figure 4 comes. That's why I think figure 4 is in there, because, again, grad student or postdoc is like, I spent 18 months of my life on this goddamn experiment. It is going to be a main figure. And to be fair, I mean, if they had just said supplementary in the text, I would have gone and looked at that supplementary figure. And I don't always look at supplementary figures, because I would have been like, really? That seems surprising. but it's very clear, like it just does nothing.
Yeah, and we are, to be fair, strong advocates. We have been in the past of publishing negative data. This is something that is highly useful, so we're not criticizing it as being a part of the main figure. It just speaks of scientific pain.
Yes, absolutely. Absolutely. Okay, and so then to try to salvage this, clearly they're like, okay, what else can we find? And so they actually hit on something which I think is kind of cool. I did not know this about CD25, I think. I mean, I may have known it at one point, but so this other molecule, also called IL-2 receptor alpha, so this is the high affinity portion of the IL-2 receptor. IL-2, for the aficionados, it's essentially like food for T cells. It's like T cells need a sort of constant supply of IL-2 to continue to function. It's a regulatory thing to say, like, we're only going to continue to grow and proliferate and do our activity if we are getting a sort of constant supply of IL-2. So therefore, withdrawing IL-2 signal is a way to suppress. And that's essentially what this CD25 is doing. It is basically taking away, it's sort of sucking up the IL-2 signal from an environment to keep it away from the effector T cells that might be trying to use it to maintain their existence. So this was another differentially expressed gene, and figure 5 is basically saying, actually, this is the thing that is doing the work here to suppress. So again, they make a mouse where they can delete CD25, and they basically show that these cells are no longer able to suppress.
Yeah,
and they sort of look at all the measures. They look at inflammatory cytokines. They look at cell infiltration. And in the absence of CD25RA or IL-2 receptor alpha, they're not able to have their suppressive function, and you get inflammation as if you had deleted the Tregs themselves.
Yeah, I wasn't looking at it hard when I had initially gone through the paper. What is their effect size here in comparison to what they had done in Figure 2? I guess it's not apples to apples, right? Although,
good question.
is this still taping the skin?
Now or is ask, I don't remember. Or is this baseline?
Because I'm just trying to get the sense of whether what we're seeing here. I mean, there's an obvious change here, right? Like it is clear that CD25, the high affinity IL-2 receptor, is having an effect and preventing inflammation at some level. I'm just trying to get a sense of whether it is the only thing, or whether it would be expected to be.
I'm trying to remember now.
Effect size, but I don't see anything obvious that could be used to directly compare those things. That's fine.
Blah blah blah. Where do they get to the results? So it's on page 8 that they're describing this. Treated Mohawk mice, topical 4-HT, and then they just do flow cytometry directly. So I think...
So we're talking about autoimmune inflammation.
I think we're talking about, in the absence of... additional stimulation...
Okay.
I don't see anything about peeling Band-Aids off. So I think it's just you remove CD25, specifically from the Tregs in skin, and all of a sudden you start getting local inflammation.
Yep.
In that area.
Yep.
Okay. All right. So then Figure 6, they're trying to get more specifically into the mechanism around the hair follicle. So the idea here is that there are human diseases that involve loss of tolerance in the skin, and they often lead to there's a couple of different things. So you can there's certain kinds of breaks in tolerance that where your immune system starts to attack melanocytes, and that's how you get
vitiligo.
Thank you. Yes, that I'm going to remove most of that silence. That's how you get vitiligo, where you're basically like destroying the pigment-producing cells. But here they're looking at alopecia, which is the loss of hair on certain sections of skin that's induced by autoimmunity. And so what they're doing here is, again, they have another mouse model where they have diphtheria toxin under the control of FOXP3, and then they're combining this with another mouse where they're doing expression of ovalbumin. So again, this is sort of a classic immunology model where you express this model antigen. Mice don't express ovalbumin, but we have very, very high affinity T-cell receptor clones that you can basically give a mouse the ability to respond specifically to ovalbumin. And so they're expressing ovalbumin in the hair follicle tissue. They're in another mouse, they've got the T-cell receptor, and they're going to transfer in T-cells from the OT1 mouse into the cell or into the mice where the hair follicles are expressing ovalbumin. Mm-hmm. And then they're going to see, and are they doing... And then they're again doing this where they can delete specifically the skin regulatory T cells. And so basically, if the hair follicle is expressing ovalbumin, even in the presence of OT1 T cells that are responding, you are not destroying the hair follicle cells. But if you remove the T regulatory cells from the environment of the hair follicle, then those T cells are able to infiltrate and destroy the hair follicle, and the hair falls out, and you get alopecia in those animals, and you get the cell infiltrates, and you get the inflammation and all of the things that you would expect. Right? So again, very nicely demonstrating that if you have, so in the previous situations where you've just got Tregs, where you're just deleting the Tregs, you don't know what the T cells that are infiltrating, what the inflammation that is occurring, you don't know what it's specific to. Yeah. You could sort of imagine like there's some antigens that are expressed uniquely in hair follicles that you're less likely to get peripheral deletion of those effector T cells, and so you just get infiltration sort of non-specifically. Here they're targeting, they're directing T cells to a specific antigen expressed in a particular tissue, and they're showing indeed you are getting infiltration of these ovalbumin specific T cells into that tissue when you remove suppression by T regulatory cells. Yep. So just sort of a nice demonstration of the I guess the T cell effector portion of this inflammation. We saw in the previous figure that it's not clear that CD8 T-cells are the entire effect, but here they're showing that at least they're sufficient to get this infiltration and inflammation in the tissue.
Yep.
In the absence of Tregs.
Tregs are sufficient to control that. Yep.
Yeah, exactly.
Yep.
Okay, and this is where they want to claim sort of the immune privilege, right? Because if you express, I think this is true. If you express ovalbumin in beta islet cells and you put in OT1 T cells, you're going to get, like, your pancreas is going to get destroyed, right?
Yep.
Even in the presence of Tregs. Is that right? I think that's a model of
I believe that that's
diabetes.
True, but I don't think we should hang our scientific hats on it.
That's fair. It's been a long time since I thought about a lot of these things. I think that's true. Not totally sure. But anyway. Okay. So that's figure 6. And then figure 7, as we've described, like this is the, like, yes, it's true in humans too. And here's where the spatial transcriptomics comes in. So they're able to get, one of the cool things about spatial transcriptomics, which is a technology that Matt has been pursuing and I am sort of helping with, is a technology where you can take a tissue section and you can, without removing cells from their tissue context, you can look at gene expression in the sort of spatial organization of a tissue. There's a couple of different technologies to do this. We don't need to go into it now. But basically, they've got these formalin-fixed tissues from 2 people that are suffering from what's called scarring alopecia, which is thought to be mediated by CD8 T-cells. And they're comparing these to healthy donors, and they're looking specifically at tissue sections from healthy skin and from skin from these folks with scarring alopecia, and they're looking around hair follicles. And what they see essentially is they see higher expression of CD28, the high affinity IL-2 receptor, that is around where they're also seeing CD8 expression and Treg expression. So around these hair follicles, you sort of have an overlap of infiltrating CD8 T-cells and Tregs that are like trying to do their job essentially to suppress. But of course, it seems like it's not working in this particular case.
That is one interpretation.
Okay, what is your interpretation?
Of what this? No, spatial technology is hard.
Yeah, for sure.
Spatial technology is hard. The version of this spatial technology is 10x's Visium platform. Mm-hmm. The challenge around this sort of thing is that the resolution is not particularly high. So you see sort of these dots, right?
Yeah.
And these dots are quite large, actually,
in
comparison to a cell. So what you're doing is you're doing sort of a segmented region of interest analysis, which is interesting and informative. And it definitely shows that you've got an IL-2 signaling pathway on, and that CD28 is present in all of these things. It is never clear in a situation like this who is expressing what, under what circumstances.
Yes, true.
What direction any of the regulation is actually occurring in, right? Like stuff can be upregulated or downregulated in a compensatory way. Now, all of this is the human data at the end of the paper that shows the relevance of the pathways that were laid out in the former 6 figures.
Exactly.
I suspect that what they're seeing is probably what they are laying out in the previous 6 figures. I would say that, if you, and I think you alluded to this at the beginning of the paper, if you came to this project with this spatial transcriptomics data.
Yeah, exactly.
Right? You wouldn't necessarily know where to take any of this, because the directionality of this kind of data is extraordinarily challenging, and it's something that, as you mentioned, we're actively working on. Spatial transcriptomics data is extremely hard to wield. It's just hard to wield.
Well, and the other thing about this that that I find a little challenging, what I really wanted to see here is I wanted to see around hair follicles a bunch of T regulatory cells expressing IL-25, or not IL-25, sorry, CD25, and no CD8 T cells. And then in the alopecia case, I wanted to see a bunch of CD8 cells and the Tregs not there, because what we're looking at in the case of alopecia is we're seeing the failure of regulation, right? We're seeing the system that they've described so beautifully in the previous paper. We are necessarily seeing that system not working. Right. And so what they're claiming essentially is that, that these Tregs are coming in and trying to suppress and not being able to, and that's why you're seeing this area of overlap of expression. It would be kind of cool if we could see, for example, that we get all these Tregs in and the Tregs are sort of separated from the T cells. But as you say, the resolution just isn't high enough for us to see the cell-by-cell count of these expression patterns. And so, I think in the context of the rest of this paper, this figure, expensive as it is, because I imagine that this one experiment was a lot of money, even though they're doing 2 alopecia patients and 2 healthy donors, right? So very hard to do anything quantitative. But what they're saying is basically we see evidence of high expression of IL-2 receptor. We see infiltrating CD8 T cells around these hair follicles that have been destroyed. And that is evidence that all of the beautiful work we saw in the mice, there's some corollary in humans. I feel like I would have, if I was this grad student or postdoc, I would have thrown in CTLA-4 here just to see, given all that work that they've done previously. But you
can't tell me that he didn't look at it.
Of course they did. Right? Of course they did.
Which means that, and
they
this
did
is,
this
I think,
experiment before they made the mouse, right? Like they
yeah,
saw
I'm sure
CTLA4
that's
all around.
true.
They're like, yeah, man,
Yeah,
CTLA4 is amazing. And then they made the mouse and it was complete BS. But
Yep. No, it's yeah. Yeah, they did what they could here. They did what they could here. What the challenge is with this sort of thing, like these targets that they have here, these images, right? Like those are the best. That's the best of what came out of this experiment, right? It's important to keep that in mind because this is sequencing based, actually, right? The Visium is sequencing based, so they had the full transcriptome here to work.
Oh, really? I didn't realize that.
Yeah, so this is spot based, in that it is like there's a specific barcode in that spot that then tags the mRNA, and then
interesting.
you sequence based off of that. So what they've got here is sort of the aggregate mRNA from within that region of interest, which is why you can't get any higher on the resolution because it's not actually spatially, it's not imaging it. It's basically capturing the RNA from that dedicated region.
I
right?
see.
And
okay.
so like you get into this and it's just hard.
Yeah,
It's just hard. Like I said, it is cool technology. It's important technology. It is important to bridge the gap between mouse and humans. All of that is true.
Yeah.
It is hard technology to wield in an effective, hypothesis-driven way.
And my initial motivation for grabbing this paper to sort of see the spatial transcriptomics angle, I basically wanted to see like what kinds of analyses are people doing, and it's very rudimentary.
Sure.
The kinds of things that they're doing. They're not doing anything like unsupervised stochastic clustering. Like, it's not quite hierarchical clustering, but it's not so different. It's just like a pretty, pretty high level of abstraction. It's not really getting into the spatial aspects other than the display. Like they're not doing any kind of spatially based statistics to quantify. So, my original motivation, as I said, was to kind of get into that, to see the kind of analyses people are doing. And like, it's useful to see that this is a Science Immunology, great journal.
And a good
all
paper.
Of the other methodology in this paper is really excellent. Like this is very, very well done stuff, and it's the spatial transcriptomics aspect of it is still, as you say, I guess it's just tough.
Yeah, and I think it's reflective, not necessarily of a failure, certainly on this group's.
I don't mean to imply that at all.
I think the field is very much grappling with how to use these data effectively. I mean, I have a weekly meeting with my grad student that's working on this, and I mean, we're literally just developing pipelines from scratch right now because you require a level of flexibility here that the pipelines that have been built for traditional single-cell transcriptomics, which they obviously use here and they use reasonably well, like those pipelines are very straightforward because you've pulled all of those cells apart, right? And the connections don't matter anymore. It's just a much easier type of data set to wield.
Yeah.
And I think we're working on it.
And that, I think, again, like this is not to disparage these folks at all, but I think that this reminds me very much, this lab is clearly, like spatial transcriptomics is not their thing. It is a technology that they are wielding in service of a different immunological question that they're pursuing. All of the other methodology is clearly like making mice, like making really well controlled experiments. Like that's clearly what this lab is good at.
Yep.
And this reminds me a lot of microbiome papers, or a lot of papers that include a figure on the microbiome. It's like you do a bunch of sequencing, you do some very basic stats using pre-built tools, and you get some results, and you try to interpret them in the context of all of your other work. That's a very different prospect than like advancing the field of spatial transcriptomics specifically, which they're clearly like that's not their what they're doing.
No, that's not the point. Not the point. But point is, Tregs in the skin, important.
Yeah.
Surprisingly, CTLA4, less important in sort of a sad way. No,
I was so depressed looking at that figure. I was like, oh.
I know. No, and you know that's like we joke that post, and I don't know if this is a graduate student or a postdoc, but like one of the jokes about graduate student training is that like you have all of these great ideas, right? And you have this project and it starts to roll out and you present it to your committee and they're like, this is great, like you're well on your way and you're going to graduate in 2 years. And then you finish your qualifying exam and you do that experiment and then it all falls on your head.
Yep.
Right? And then your PhD is about like what happens after it falls on your head.
Yeah, no, this is like the figure 3 slump, or I mean year 3 slump, basically. Slump. It's just exactly. Like nothing works. I mean, you got to imagine that this was like this. I don't know how long it takes to make a mouse in 2025.
Not long, actually. Not long.
This was published in January, so in 2024. It doesn't take long, but it's not like you place an order and you get it in a week, right? Like it's work. Like you got to do the breeding, you got to like separate the cages, you got to do the crosses, and so it's non-trivial to set up all of this stuff, and then doing the actual experiments. I mean, they're doing the painting, they're doing this is weeks of work, and I gotta think that this person, whoever was doing this experiment, the whole time. They're imagining the figures in their head, right? Yep. Like, if they're anything like me, like you're doing the experiment, you're pipetting the stuff, you're painting on the skin, you're shaving the mohawk, you're thinking the whole time, this is what this figure is going to look like. It's going to be so great. It's going to be figure 2 in my nature paper. It's going to be like, it's going to be grand. And then they get this data and they bring it to their PI. And depending on the PI, I don't know this particular PI. I can tell you in my graduate student career, I think I've told the story before, a long-standing project that had tons of potential. We were very excited. I did this one Western blot that told me that it was an artifact. Like the whole thing. It was complete BS. And like I remember like the fear that I had walking into John's office with that Western blot. Like it just hurts. And scientific misconduct is never justified, but in those moments you can sort of understand the mindset of people that use Photoshop to fuck with their Western blots, like you can, because it hurts so bad to have that data that shows that all of the work that you've done for 6 months is wrong, or is essentially useless. They managed to use the data, which is great. I mean, I think figure 4, I think I'm really happy they included it. But I'm just sort of imagining the emotional mindset of that person walking into their PI's office with that data.
And I would say that a hallmark of a good scientist, and that, it doesn't always happen, but a hallmark of a good scientist is somebody being able to point back to a project that they just simply had to bury because it turned out it wasn't what they thought it was, and it was really exciting right up until the moment that it wasn't, and something turned on its head, and rather than beat it to death and continue to press along that way, like the ability to take an objective look at it, document it effectively, and then put it to bed, like that that is something that really should be celebrated in science more.
100%.
It is hard to do that. It is hard to do that.
That meeting that I had with John with that data, that's like a defining moment in my career.
Yeah.
His reaction to me showing him that data, I feel like that's almost like an origin story. It's like I walked in with the data, he looked at it, he looked at me, and he was like, all right, so what's our next project? What do we do next?
Well,
this is clearly dead. Move. I was expecting him to be mad. I was expecting him to be like, well, do this other experiment, all this stuff. And he just he took it for what it was.
Yeah.
We buried it and moved on. And that reaction to me was so formative in terms of the way that I think about the task of doing science, which is like there's a lot of failure involved. And anyway.
No, we've talked about it before, but like disaggregating your own self-worth from the validity of your scientific ideas is a very, very surprisingly hard thing when 100% of your life for 6 years is spent trying to gather data to graduate from a program, right? And that is a hard lesson, and not everybody learns that lesson, unfortunately. And for those that don't learn that lesson, it is a hard path to be a scientist because then you live and die by whether those projects are working. Ooh, that's tough.
Speaking of which,
yes,
Jay Bhattacharya. I'm just kidding. We're not going to do that. We're not going to do that.
Nope. This has been
Audio Immunity. Episode 40. And, we're trying to churn these out twice a month. Mm-hmm. If you want to support our efforts here, patreon.com/audioimmunity. Audioimmunity is spelled AUDIOMMUNITY. You can also find us at audioimmunity.org. You can find us on YouTube now, youtube.com/@audioimmunity. We're getting not many views on YouTube, which makes me feel a little bit less bad about picking like the weirdest face that we're making for the thumbnails.
Yeah.
But you should come watch us on YouTube.
Yeah.
You should come listen. You can find our feed on Audioimmunity. If you subscribe on iTunes or like really any podcatching service, please leave a rating and review. Like and subscribe on YouTube. It always helps. And share us with your friends. If you're listening while you are patiently painting 40T, whatever the heck it is, on your Mohawk mouse, really excited about CTLA-4 and its effects.
Yeah.
Imagine what some other graduate student or postdoc doing the same experiment could be listening to while they're doing their destined to fail mouse experiment.
Right. Give the gift of Audioimmunity.
Give the gift of Audioimmunity. Exactly. And I think that's it. The music at the beginning and the end of the show is composed by Rachel Reinick. And next episode for Really Reals, Kate is going to be back. We're going to be talking about a paper that we tried to record on several weeks ago that we were not able to do. I have the in to squat in this office for recording now, so my internet works. I'm already 99% uploaded. Life is good.
And you've got nice art for those of you watching on YouTube.
That's true.
Added art.
Yeah, it's true. It's beautiful. You should come look, come see on YouTube. And we'll see you next time.
Sometimes